we are in the basics of genetics, which is useful for 10th ICC as well as 12th CBSE biology. So if you find these videos useful, please subscribe to the channel and like the video. Kindly share with your friends as well so that they can learn along as well. Alright, so we have already done few videos on the basics of genetics covering all the important concepts. Today we are taking up Mendel's experiments and laws of inheritance. As you all know, there are 3 laws. We will cover all the laws in 2 separate videos. So this is video number 1 in that. So first and foremost, who is Gregor Mendel? He was Austrian monk who studied basics of genetics, and therefore we call him the father of genetics. He studied pea plant, therefore you are supposed to know the scientific name of pea as well. Paisum sativum, while writing, make sure that you write it as 2 separate words starting with the capital P for the genus and small s for the species. Underline both of them separately. Please don't forget all these small details. He studied 7 contrasting characters, and this table you're supposed to know without any error. Meaning, which were the characters he studied and what are the 2 traits for that. So, what do you mean by character? We should know, isn't it? So, anything inheritable from a parent to the offspring, anything that you can see, we call that as character. So, for example, best example to take is the height. If you take height to be the character, the 2 traits for that are tall and dwarf. Tall and dwarf. It's not written here, but that is what is meant by character and trait. So, for height, the character is height and the 2 traits are tall and dwarf. The same way for so many other characters he studied, 7 of them, all the 7 you should know, and under the 7 which all are the dominant ones and what is the recessive trait you should know. Because whenever a question is given to you to work out like this, they may not tell you which is the dominant one. Alright, coming to Mendel's experiments. So what Mendel did was, he studied all the 7 characters, but he didn't do all the 7 at once. What he did was, he took one character, any one among them, and he studied. Alright, so when you study only one character for a plant, then you call that as a monohybrid cross. So what exactly is a monohybrid cross? Study of inheritance of only one character at a time. Here, first example we are taking is height, how Mendel did. There are 3-4 cases given in your textbook. All the same, just height is the easiest one to comprehend. That's why I have taken height. Alright. So what did he do? He took pure breeding tall plant and crossed it with pure breeding dwarf. So what exactly is meant by pure breeding? The word pure breeding means homozygous. Please note that down. It means homozygous. And he took tall plants. So we have already learned how to name any genotype. Correct? So every gene will have 2 alleles to it. Correct? So in this case, we know that the alleles are homozygous. That's why we have taken it to be capital T and capital T. If you have any confusions in that, please type in the comment box. I will explain this further. Alright, and then he crossed it with a pure breeding dwarf. So we know dwarf is a recessive character. Whenever we have to make the genotype of a recessive character, we don't go and take D and D. That is one error that you will make. Please don't do that. That's not the convention of naming. How do we name? We have to take the first letter of the dominant trait, that is T. For expressing dominance, we have to write capital T or uppercase T. To express recessiveness, we have to use smaller case of the first letter of the dominant character. That's why we have a T and T here. Alright. Now, once this cross is done, how did he do the cross? That also you should know. So, from the tall plant, he took pollen grain and dusted on to the stigma of the dwarf plant. Alright. So, that is how crossing is done. And if you are doing this process, the dwarf plant we have to consider as the female, isn't it? And this we have to consider as the male. We can consider either way, it doesn't matter. In this case, we are taking tall to be male and dwarf to be female. We have to take out the pollen grain from here and then dust it over here. So now, before doing that, we have to make sure that the pollen grain of the female plant itself. So as you know, if this is the stigma, and already we know anthers are right next to it itself. So there is a chance that the pollen grain will come and fall on the stigma. So in order to prevent that, what he did was he removed the anther of the female plant, and that is called as emasculation. So that also, just for your information, you should know. Alright, so how is the crossing done? Crossing is done by dusting the pollen grain from the plant we are going to consider as male onto the stigma of the female plant. That's what it is. Alright. Now, once that is done, we should know how to make something called as a Punnett square. Alright. So, what result did Mendel get? Mendel got by crossing tall plant with dwarf plant, he expected some tall and some dwarf, obviously, as the progeny or the next generation from the plants. He expected some tall and some dwarf, but that is not what he got. Instead, he got all tall plants from the next generation. So, next generation is represented by F1. F1 stands for filial. What is filial? Filial means son or daughter. So F1 is the first filial generation or the next generation. Okay. So in order to find out what results he got, we need to know to make the Punnett square and solve it. So what do we do for that? We need to make a box like so, which I have already drawn on it, and we have to take one allele at a time and place it over here. So these are to be considered as gametes. So before filling the Punnett square, what you have to do is take the first allele and put it inside a circle, and that we call that as a gamete. And this is the next gamete. We have to cross it with these two gametes. Alright. Now fill this over here and this over here. This T will go over here and this I am going to place it here. Now how do we make the cross? The cross is made by this and this and this. Please follow my cursor. This and this and this and this and this. What do we get? We get the results you have to write down. Alright, results, the genotype of the result of F1 is capital T and small t. What is capital T and small t? This refers to tall. What kind of tall is it? It's heterozygous tall. This concept we have already covered. That's why I'm not discussing it again. Alright. Now, he got all tall. So therefore, he expected that in the next generation, everything should be tall, isn't it? Because he knew that first generation all are tall. So therefore, second generation, how will anywhere dwarfs come? He thought that. And he crossed he made the F2 progeny. How did he make the F2 progeny or the next filial generation? By self-pollinating F1. How to self-pollinate F1? That is very easy. If one of the flower you take, its stigma will be there, its anther will be there, isn't it? Take out the pollen grain of this and dust over here. That's it. Self-pollination done. Alright. So that is what he did for the F1 generation. So let's say he took this and then from the seed of it, he grew the plant, and from the plant he did F1, he crossed self-pollination he did to make F2. Alright. So how do we make gametes before crossing? So we know that F2 is equal to F1 crossed with F1, isn't it? F2 is equal to F1 crossed with F1. What are the genotype? What is the genotype of F1? It looks like this. And you're crossing it with this. So before filling it over here, let's make gametes for this one. Gamete number 1, gamete number 2. Other one also, same thing. because F1 is basically its own. So now we are going to fill this T over here, this T over here, this over here, and this one over here. Now let's cross. How do we cross? Let's please follow my cursor. This and this together you get. This and this you get. This and this you get. Why am I writing the capital T first and not small t? Why can't I write this one as this? I cannot write that. Okay, it's because whenever we are writing the genotype, the dominant allele to be written first. Now, what did you get from this? The results. In the results, you notice that there are how many tall plants? 1, 2 and 3. Let's write it down instead of me saying it. Let me use another color. So, what do we get? We got TT. This one, we got 1. This and this. Capital T, small t. How many we got? We got 2. And this one, small t, we got 1. Correct? So the genotypic ratio, genotype meaning this genetic constitutional ratio becomes 1 is to 2 is to 1. 1 is to 2 is to 1. However, if you notice, this and this are tall itself. Correct? They are all tall. Capital T or capital T small t, whatever the condition is, since there is a dominant allele over here, this is going to be tall. Alright. So how many tall we got? 2 plus 1. 3 we got. 3 tall and 1 dwarf. So what will be the phenotypic ratio? What is phenotype? Phenotype is what you see. Correct? So this we have to fill. The phenotypic ratio becomes, phenotypic ratio becomes 3 is to 1 and genotypic ratio is 1 is to 2 is to 1. Now, what was the, what cross was it? It was called as a monohybrid cross. Now, from the monohybrid cross, Mendel came up with one law which you have to study. And this law states, out of a pair of contrasting characters present together. What are they? In this case, what were the contrasting traits? Instead of character, take it to be trait. It's much easier to understand. What were the two traits? One was tall and one was dwarf. These alleles were together. Contrasting alleles were together. One is able to express itself. In this case, which one is expressing itself? This is expressing itself while this is getting masked. Right? And that is law of dominance. What is law of dominance? Out of a pair of contrasting characters, capital T and small t together, one among them is expressing itself and that is called as the dominant one. This one which expresses itself. And the one that gets masked is called as the recessive character. Alright, so that is law of dominance. In the next video, we will understand the next two laws using dihybrid cross.